Hi, sir. Biochem Cadet 1. Biochem Cadet 2.
Sir, class, I'm Kevin Pack, and I'm here to brief the HIV1 protease. So in terms of structure, as you can see here, sir, the HIV1 protease is a dimer. It consists of two monomers with 99 amino acids each. So each component has one aspartate amino acid group, which means that it's a dimer aspartyl enzyme. So in total there's 198 amino acids forming the primary structure of this enzyme. So in terms of secondary structure, obviously there's alpha helices right here and right there, and majority of its secondary structure are beta-plated sheets. One interesting component of the secondary structure is that up here are two flaps that consist of two beta-plated sheets each and a loop. And so the whole function of that is after the binding event occurs, the two flaps enclose around to help the reaction process actually complete. So in terms of tertiary structure, main forces that stabilize the tertiary structure are entropic forces. So the hydrophobic interactions inside and the hydrophilic parts outside stabilize the tertiary structure. The forces that stabilize the quaternary structure are mainly van der Waals interactions and disulfide bonds. And as you can see here, the disulfide bonds aren't actually, the disulfide bonds don't link up the two different monomers together. They stay inside of the same monomer, but they are positioned so that they cross over. So even though the disulfide bonds aren't joining two separate monomers together, the fact that they're crossed over kind of increases the stability. Hence, it stabilizes the quaternary structure. So in function, the function of the protease, the function of all proteases are to cleave peptide bonds. So the most common protease that people think of, they break up peptide bonds, they break up proteins for digestion purposes. The HIV1 protease is different because it actually breaks down the viral proteins of the HIV virus, and it makes up viral proteins that help in the replication process of the HIV1 virus. So there's a picture right here, just a quick diagram. So the peptide bond is the bond between the carbonyl group and the nitrogen group, and it basically cleaves this through the process of hydrolysis. So water is added, and through nucleophilic attack of the water on this protein, the enzyme helps cleave this bond. Typically, protein hydrolysis is thermodynamically favorable, but it takes a really long time because of resonance that occurs all through here. And so the purpose of this protease is to speed up the process and to make the hydrolysis happen. So activation and inhibition. So first, the HIV1 protease exists inside of the HIV1 virus, obviously. But initially it exists in a dormant state called the GAG-pro-pol polyprotein state, and a whole bunch of viral proteins along with this protease exists inside this polyprotein. But as soon as that polyprotein is activated, this protease also activates and starts cleaving bonds. The actual turning on system is not really known, so it's still under study. How the GAG-pro polyprotein complex is activated is not known. But to stop this HIV1 protease, there are four main inhibitors that are being studied. The sequinavir mesylate, the ritonavir indinavir sulfate, and the nelfinavir mesylate. There's a picture here. So these two star clusters here, as you can see, like on the slides, those two stars symbolize the aspartate amino acid groups, which essentially are the active site of the enzyme. And here's a picture of what it looks like when some substrate is actually bonded to the aspartate groups. And then here's when the two flaps we talked about initially, they close over the water and the substrate to actually make the hydrolysis happen. So the ultimate importance of this is that trying to use inhibitors to stop the HIV1 protease to cleave peptides can be potentially used as an anti-HIV therapy, because ultimately if the HIV1 protease can't cleave proteins to make tools to make more copies of itself, then ultimately it stops the HIV infection from spreading, which is the key here. So the inhibitors that I mentioned are being used to help stop the HIV infection from spreading by stopping the process of protease altogether. But mutation is also a growing problem. The HIV is like just any other virus. It constantly mutates. And so this mutation leads to drug resistance. So the inhibitors that I mentioned before are starting to not work as efficiently for HIV than it has before. So one specific mutation that stops the protease from performing, or stops the inhibitors from doing its function, is the G48V mutation. It essentially induces more van der Waals forces among the A53 Phi and the A48 Val components of the enzyme, and by doing this, it increases the interaction between these components and the inhibitor, and in that process, it actually rotates the inhibitor by 30 degrees, and even that turning can actually stop the inhibitor from actually binding onto the aspartate groups on the active site. So it overall stops the inhibitor from actually binding, which causes the protease to not stop functioning. So the HIV1 protease will still continue to cleave proteins and make more tools to amplify itself in the inside of the host. So fortunately, that's not the only way to stop the HIV1 protease. Cysteine modification is one way that the HIV1 protease activity can be controlled. There's two ways of controlling the cysteines. The first is oxidation by the ions. So, for example, zinc here, it binds to the cysteine groups, and by binding to it, it reduces the activity by half. Another way is by adding cellular thiol glutathione. So if you add this component to a cysteine 95, it actually stops stops the entire process altogether. But if you add this on cysteine 67, the protease activity is stabilized. So essentially, the first way on cysteine 95, it stops activity, while adding this to a cysteine-67, it stabilizes and it encourages the activity. So there's just two ways to just control the level. Obviously, from a health science standpoint, the goal is to stop or to stop the protease activity. So these are my references and questions. Yes.
How long does it take to develop one of those inhibitors?
Develop?
Do you have any idea of how long it would take?
Well, it takes extensive research, obviously, but the four that I mentioned were the only ones that were made. I don't know how long it took to actually make each drug, so it probably takes a while. I mean,
so the idea is
really being
to stay ahead, right?
Yeah.
Okay.
Yeah, especially so staying ahead is a really important concept here, because the viruses tend to change a lot and catch up faster than our drugs can obviously stop them, and so it's a race essentially. So using the inhibitors are not really they are still the primary focus on stopping the protease activity, but there has to be alternative means, and this cysteine modification are one way, on how to stop it, because soon all inhibitors that we have in our arsenal right now may not be effective 50 years down the line because of mutation.
Going back to, I think, dimer, the dimer portion, I may have missed this, but are they equivalent, or do they each have a different,
Yeah, so they're exactly the same.
Okay.
So both components are exactly the same, and they just link together down here by sulfide linkages.
Okay.
So both has 99 amino acids group, including the aspartate residues as the active site.
Okay. 'Cause I was just wondering if they had specific, if they were
No,
different
they're the same,
specificity.
yeah.
Okay.
They're split down the line. They're
Just like a capsular or the other ones.
Yeah.
That's cool. Alright.
All right. Thanks, sir.